that they usually had other professions, right? So I believe Robert Hoke was also a priest, and Anthony Van Leeuwenhoek was a fabric merchant, right? And so science was kind of a side gig or an outside interest, all right? And now we see that we've become a lot more specialized. I'm just responding to a chat. And when you guys have questions, you can just ask if you're comfortable with that, or you can type into the chat. You can make it a group question, or you can make it a private chat, whatever you're more comfortable with. When it comes to microbiology, one of the biggest obstacles to overcome was essentially this idea of spontaneous generation, all right? Microbes came from nowhere, or, right, it was actually just bad air or bad water that was causing infection. And in some cases, there's a little bit of truth to that because the microbes were in the air or the microbes were in the water, right? But our understanding was very, very limited to start. And so these individuals here, I'm not necessarily expecting you to remember them, but they do a kind of, they each do a series of experiments that are looking at where microbes come from, all right? And so John Needham, let's see, can I exit full screen? No? Oh, okay, I guess I'm stuck here. Oh, there we go. Oh, that's a totally different lecture. Hold on, sorry. Small technical difficulty. Where did that go? Okay. There we go. All right, you guys can see that again? Okay.
Yes.
Good. Thank you. Sorry about that. So what you see is some individuals trying to figure out what's going on. So you have John Needham, and he basically comes up with this idea of, all right, I'm going to boil a broth, right? So think about it, something like a chicken broth or beef broth, all right, or vegetable broth, something that seems nutritious. I'm going to boil it. I'm going to plug it, all right, and then theoretically, if there's something in there, boiling it would have killed it, and nothing should happen, right, if microbes don't spontaneously appear. So John Needham boils his broth, plugs it with cork, lets it sit, and then things grow. Why is that a problem? Olivia, what do corks have in them?
Like other pieces of the cork could fall into the broth, and so it could look like material that wasn't actually there.
Okay, so if it's a bad cork, all right, okay.
Cork's also porous.
Yes, corks are porous. And so both of you, I'm sorry, I'm going to have to turn into the president's office, because at Lee, you shouldn't know what cork is. Okay? Because that usually plugs wine, and that's a no-no. So you're busted.
That's fine.
That's okay, good, perfect. I'm just kidding, Olivia. I don't know you. Trent knows me. He doesn't take me seriously. That's fine. But, right, corks are porous. And so although our good friend John, right, thought he was totally keeping the air out, it turns out he wasn't, all right? So another, we'll say, kind of contributor pioneer to microbiology was Francisco Redi. And if any of you have heard the experiment where you take a piece of meat and you put it in a jar, you leave the jar open, right? Flies and other things come to the jar, right? We now know that things start to pop up and appear on the meat because those insects are laying their larvae on it. They didn't have an understanding of that then. And so they would say, oh, we're having spontaneous generation of these bugs. So what he did is he took 2 jars, one that was open, had meat in it, one that was closed, and he put a piece of kind of porous felt on top. And so the aroma from the rotting meat would waft out. The flies and other insects would land on that piece of kind of porous velvet, and they would lay their larvae there. And then you would see bugs start to grow or form on the outside of the jar, right? So although not directly related to microbiology, it was still important, right? And kind of this idea of, hey, things don't just spontaneously pop up. Lazzaro Spallanzani basically repeated John Needham's experiment, but did a better job of sealing his flasks. And so he actually did not see microbe growth after boiling, and we'll kind of talk about an exception to that in just a second. And then my main man, my boy, Louis Pasteur. So if you've heard of pasteurization, he also was a pioneer in vaccination. We'll talk about him a little bit later, right? There's the Pasteur Institute in Paris, all right? So kind of Pasteur and this other guy, Robert Koch, are just 2 of the biggest figures in kind of modern microbiology ever. And so what Pasteur did is he made this cool thing called the swan neck flask. And what he did here is very similar to the people before him. He put broth in here. He boiled it, all right? And as it escaped, right, or as it boiled, right, you had gas and moisture that would leave, right? That moisture in the curve of this flask, right, what we now know, or what Pasteur figured out, trapped microbes from the air in them, right? But they couldn't get to the broth, and so they wouldn't grow. And so all you'd have to do then is turn the flask on its side. So this trapped stuff would come in contact with your broth, and then you'd have growth, right? So from this, he was able to figure out that microbes were not spontaneously generating. They were actually, in this case, coming from the air, all right? So all of this stuff sounds very simple and very basic to us, but at the time, these discoveries were definitely revolutionary, and it took a long time, right? In some cases, hundreds of years to convince people that things weren't just spontaneously generating. Now, as always, right, we kind of see people confirming the results of other scientists, which is awesome. And sometimes we see things that are slightly different. And so immediately we start to see some conflicting results with Pasteur and some of the others that have come before them. Specifically, some broths, all right, are still able to support microbial growth after extensive boiling. Okay? There we go. And so in this case, right, we have John Tyndall, who speculates that there are resistant organisms that can survive boiling. And not long after, right, a scientist named Ferdinand Kohn actually discovers something called an endospore. Endospores are primarily found in 2 genera of bacteria, Bacillus and Clostridium. And basically, they are a bacteria seed. And we'll talk about these more this week. But these little seeds, even when the bacteria dies, right, are able to later become fully grown, dividing, happy bacteria. They're resistant to heat. They're resistant to radiation. They're resistant to antibiotics. They're resistant to dehydration, all right? And so they're really good at allowing bacteria to survive. We'll talk about this in a little bit more detail. Those of you that are in the healthcare field have probably heard of C. diff. Clostridium difficile is the full bacteria name. It causes life-threatening diarrhea in elderly patients. They're not able to regulate liquid intake, right? They become dehydrated very rapidly, which eventually can lead to their death. And what doctors do is they treat these patients with antibiotics, and oftentimes the patients respond really well, but they will have a reoccurrence because antibiotic treatment kills the bacteria, but the endospores are resistant. So you think that you've helped a patient out, they're doing well, and then those endospores essentially, and we'll talk about this more, but hatch into fully grown bacteria, and then you see a reoccurrence of C. diff. All right. So again, we'll talk about that more. Kind of two other prominent figures in microbiology are Jacob Henle. So if you've heard of the loop of Henle, it's actually an inoculating tool that's used in a lab that we would be using if we were there in person. But Jacob Henle and Robert Koch are basically really essential to this idea that most of the diseases that the world knew at that time actually had a link or were being directly caused by microorganisms. Again, their ideas were met with a lot of resistance, and if you actually go and read the literature, there's speculation that Koch actually stole a lot of ideas from Henle. But Robert Koch is essentially is responsible for something called Koch's postulates. And this is still kind of determined, or is considered the gold standard for determining if something causes a disease. Now, because of things that we know, these 4 postulates, there's been some slight modifications to them, but they still generally hold true. All right. This idea that the same organism must be present in every case of disease. And so if Elena, Sarah, Lauren, and Anna all appear to have the same disease, and I take their blood and I try to culture it, and from each one of them I get 4 different microorganisms, well then, right, I need to keep searching because clearly right, something's wrong there. So the same organism or the same microbe needs to be present every time. All right. The organisms have to be able to be isolated and grown in pure culture. All right. And then the isolates have to be able to cause disease when inoculated into a healthy, susceptible animal, right? Not a person. That's a no-no, right? We have laws and rules that protect people from things like that, but we'll actually talk about some examples of that throughout history that are actually pretty important in our understanding of pathogens. And then once you infect an animal, you have to be able to re-isolate it, all right? Some of the exceptions are now when it comes to things that are living in your gut, right? I talked about the gut microbiome in that video. The gut is an environment that we can't simulate perfectly, right? But through modern, I guess we'll say, identification techniques, and we'll talk about these in more detail, even though we can't grow them, we know that they're there, right? So in that case, we might not be able to get a pure culture of them because we don't really know how to grow them well, but we can still associate them with the disease, right? So there are exceptions there. We'll go over this more later, but how do we name microbes, right? So some of you have probably done phylum kind of analysis, or not analysis, but you've looked at these before, maybe in your kingdom, phylum, class, order, family, genus, species, right? So typically we're interested in naming something by the genus and the species, right? So in this case, E. coli down here, just an FYI in the future, and I'll point this out again. When you are referring to an organism by the genus and species, right? The genus, the first letter is capitalized. The species is a lowercase. If you're typing them, you either italicize them, or if you don't want to italicize for some reason, then you have to underline. And so that's just kind of a little pet peeve, but I'll point that out a little bit later. All right. So as far as microbiology, what are we typically thinking of, right? And I think the answer would probably be we're thinking of pathogens. But remember, I said for the most part, pathogens are actually a relatively small representative of microbial diversity that exists out there. All right. So all pathogens are microbes, but not all microbes are pathogens. Now, this chart is dated. You can see at this point quite a bit, but in general holds true. And so I know, maybe a lot of you are like, well, yeah, there's a lot of COVID, COVID related deaths going on. That would still at this point be a relatively small blip on this, radar. It might be getting bigger. We'll have to see when it's all said and done. But it would not at this point be a gigantic spike yet. What you can see here is on the y-axis, you have the death rate. I can't remember if it's per what, but that's fine. You see year down here. This is the United States. And so you can see throughout kind of the early 20th century, we have a couple different things going on. Here you see the introduction of health departments within states, all right? And so you start to see a decline, right? You then see chlorinated water being introduced, right? So kind of what we think of as modern plumbing becomes more common. And so a decline in disease there. You have a spike. So this is the 1918 influenza pandemic. You've probably heard this referred to multiple times during the corona or COVID-19 pandemic. It's estimated low end 20 to 40 million people died worldwide from the influenza pandemic. There's actually really cool examples even in American history. I can't remember what time of the year it was, maybe in 4th of July parades, but like St. Louis decided to cancel them and have people stay inside. And Philadelphia decided to have their 4th of July or parades. I think it was 4th of July. I can't remember exactly what the celebration was. So Philadelphia went ahead and had it. And then Philadelphia actually became one of the hot spots for influenza. And so it's actually a great example of the benefits of social distancing and isolation can be, right? But this was a really big spike. You can see, and we might mention this later, the last human transmission, documented transmission of the plague occurred here, right? So you see plague is no longer a problem. It actually was a problem in the United States even in the early 20th century. A couple people get the plague every year still in the United States. Primarily it takes place in the national parks out west. And a lot of times it will be stray animals that live in the park, have fleas. Hikers or campers pet those stray dogs typically, and then the fleas jump and bite the patrons and they get the plague, right? So if you're at a national park out west and you don't know the dog, I wouldn't pet it, right? But to each your own. We do have antibiotics that take care of it. I just feel like it's something that's a little harder to diagnose now because a doctor's first thought when you come in is not, oh, Rachel has the plague, right? That's not usually what they go, but we can treat it now. You see 1942-ish is when penicillin is first used. So the first antibiotic. It's actually within a few short years that we have we start to notice antibiotic resistance. So it doesn't take long. That'll be a big topic that we talk about. You see here, 1950s, the polio vaccine is introduced. We did have vaccines before this, but the polio vaccine is kind of one of the first big mass-produced vaccines. And then you have passage of the Vaccination Assistance Act. All right. You start to see laws where schools require vaccination. We live in an interesting time now where that's not necessarily honored anymore, all right, or you can claim freedom of religion from getting vaccines, and we'll talk about that, right? Because we will spend some time talking about the anti-vaccination movement because it's important for preventable diseases. We also see or are seeing a spike in anti-vaccination rhetoric during the COVID pandemic, right? So really briefly staying on kind of the pathogen front, what you see here, these are called emerging infectious diseases. And typically we'd say that's something that's appeared within the last 30 years. You can see that there's a little bit of leeway there. Some of these things appeared, more than 30 years ago. And when we say emerging, right, this is not, again, like they just popped out of thin air. But because of our lifestyle, because of global expansion, right, we are encountering pathogens that maybe we never encountered before. And so you see, even with the COVID outbreak, and actually these might be some of the videos that I'll show you, but if you've watched anything on them, right, they took place in Wuhan, China, in a wet market. And in these wet markets in China, and other countries have the wet markets, but China is one of the largest, has the largest wet markets in the world, and basically have a bunch of different animals that are brought in, right? Typically exotic animals that are brought in are housed closely together in these wet markets. You have all different animals that are kind of mixed together in very tight conditions. You also have the slaughtering of the animals in the same facilities and same areas. And so it allowed, or what we speculate is that it allowed transmission of the COVID-19 virus, right, from bats to humans. And I can't remember does anyone remember the other animal? It kind of looks like a little cat monkey thing. I'd never heard of it before. But anyway, right. So it allowed transmission of this virus. And so that's some of the things, that we're seeing. My other favorite one, bovine spongiform encephalopathy, right? Does anyone know what the other name, what the common name for that disease is? Allie, do you know?
No, I don't know.
No. Has anyone been to England?
I have.
You have? Who is
Yeah.
that?
Tommy.
Tommy, I can see the T, but I couldn't see you. What's one of the things that, well, I mean, you can eat it, but that some people maybe caution you to eat when you're in England? You know?
Is it like a dough product? Like, I don't know. Like, is it dough? Like scones or like biscuits or like what? Like,
No,
is
not
it
that I know of. What's wrong with their scones and biscuits?
no, like I'm like asking like what category of food is it? Is it like a dough product or like
Meat. How about it's a meat product?
meat?
Yeah.
I have no idea. I don't know what it could be though.
What's your favorite meat?
My favorite meat is like chicken. I love fish too.
Tommy, you're killing me here.
What?
That's fine. So what I'm trying to get out of Tommy, right, is that it's actually beef. If any of you have ever given blood at like the American Red Cross, before you give blood, you have to go through like this sheet, and they'll ask you like where you've been and how long you've been there. And one of the places that actually could prevent you from giving blood is if you've spent a prolonged amount of time in England. And that is because, right, England had this bovine, right, cow, right, spongiform. So if you've heard of mad cow disease, it's not quite as big now as it was when I was younger, probably before you guys were born. But it's actually one of those prion-associated diseases. And the reason that mad cow disease spread so easily in the United Kingdom, and we did have some cases in the United States, is because farmers looking to save money, right, what they would do is when a cow was slaughtered, right, and we took away all of the meat products that we used, they would then take the leftovers of the cow, which included the brain, and grind it up and basically make a protein meal, and they would feed it to the other cows. All right, so they're basically using the dead cow leftovers to feed their live cows, right? And this perpetuated the spread of mad cow disease, right? And we'll talk about that a little bit more, but it has basically causes holes to form in your brain. And, that's not good, right? Agreed? Good. These are actually just a couple examples. You can click on these on your own. I think the links are still active. I don't know if you can access all of it, but these are actually a couple examples of current events. So if you guys are like, I have no idea what he's talking about or where I should be looking, this kind of helps you out. This article is about plague in the national parks. This article is actually about Alzheimer's disease. So if you know anyone that's been affected by Alzheimer's, what some scientists have found is that they think one of the causes of Alzheimer's is actually a bacteria that infects the brain, and it divides really, really slowly. And so it's hard to detect. And what this group did is they actually started treating patients with antibiotics and noticed an immediate improvement in a lot of their patients. So it's kind of a cool thing. And this article actually talks about using E. coli to detect landmines. And basically, what researchers were able to do is spray E. coli over a section of land where they thought landmines were present. And landmines are a lot of them are made of a similar material. And basically that material starts to decompose and leaks into the air and soil. And these E. coli that they designed metabolized that byproduct of the mines that were slowly kind of breaking down. And it would cause the E. coli to fluoresce. And so you could then shoot lasers kind of at the ground to make the fluorescent E. coli even brighter, and it would tell you where landmines are, right? And so, I like this example because it doesn't have to be disease related. It could be food related. It could be waste related, right? One of the kind of things that we're interested in microbes for is how do they help us get rid of waste, right? And so some of them had enzymes that can actually break down plastics, right? So there's a lot of different ways you can go about this. You don't necessarily have to keep on looking at diseases, right? So as far as what are microbes doing for you, all right? And these are a couple big things, and we'll talk about them, right? Nitrogen fixation, carbon fixation, photosynthesis, decomposition, fermentation, bioremediation, and digestion. Can anyone tell me what nitrogen fixation and carbon fixation are?
I know that carbon fixation is converting like atmospheric carbon into like organic carbons that like plants can use and like fuels. Nitrogen fixation is converting atmospheric carbon into like I think like ammonia or something like that. I'm not so sure.
Okay, good. That's perfect, right? So Tumi's right. So basically, you're taking these elements out of the air, right, and converting them into something that's usable, right? And so carbon fixation, right, you take atmospheric carbon, you convert it into organic carbon, right, that can be used to build sugars, fats, nucleic acids, and part of proteins, right? You can't breathe in carbon and convert it into something that you can use. You need bacteria for this. You also need plants, all right? So those of you, and I realize it's not all of you that maybe took Bio 2 or took bio in high school, right? This is one of the things that we talked about. Nitrogen fixation, Tumi's right there also, right? You're taking atmospheric nitrogen and you're converting it into a form that's usable. Now, in this case, plants, right? With the or plants that are being aided by bacteria, right, can take nitrogen and convert it into ammonia or ammonium, right? The plants take that up. They can then convert it into a form of nitrogen that's incorporated into organic molecules. we then eat plants, or we eat things that eat plants. And that's how we're getting our usable nitrogen and our usable carbon. Tumi, can you remember, and this is kind of a general question, what do nitrogen fixation and carbon fixation require a lot of?
Um,
Do you remember?
okay, for carbon fixation, so I did a paper and he had like it required like Rubisco, which is an enzyme, but I don't know if that's what you're asking for.
No, that's not. So Rubisco, you're totally right. Right. So Rubisco is an enzyme that is necessary for carbon fixation. We'll talk about that later. It's the most abundant enzyme in the world. So if you're capable of carbon fixation, what else are you probably also capable of that's on this list?
Photosynthesis.
Photosynthesis, right? Perfect, right? So nitrogen fixation and carbon fixation require a lot of energy. And so oftentimes, not all the time, but often, right, they're associated with one another, right? You cannot generate enough energy to fix nitrogen and to fix carbon and to do everything else that you need to do, right? They require a lot of energy. And so photosynthesis, right, is a great way to get this energy. Let's see. Megan, can you remind me of the very, 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 very basics of photosynthesis? What's happening? How would you define photosynthesis?
Like plants converting energy from the sun into like sugars or other usable.
Perfect. I'll take it, right? And so plants is good. What I want to shift away from just a little bit is that plants do this a lot, but we do have some bacteria that also do it. And so Megan was totally right though, right? I would just define it as converting, all right, radiant energy into ATP, right, that can then be used, right, for doing these different things, right? One of the ones we think of is carbon fixation, all right? There's a lot of radiant energy out there, right? So this is a very good way, all right, to get the energy that's needed to complete these two processes above it, okay? Also, one of the reasons, not that we talk about it here, but you know that solar energy is worth investing in. The sun shines a lot, and so it's a great energy source for us to use. Decomposition, all right? If you have been driving down the road, hiking in the forest, right, and you smell something nasty, it's probably because it's decomposing. If you've seen leaves, right, that look like they're breaking down, right, bacteria and, right, fungi help with decomposition. Decomposition is essential because it is returning, right, a lot of nutrients or elements into the environment where they can then be absorbed or taken up by other organisms, right? Ooh, fermentation. What is fermentation for? Laura, what's one of your favorite things that's fermented?
Kombucha.
Ugh, I knew you were going to say kombucha. All right. So if any of you like to drink kombucha, if any of you like, I have on this page cheese. If any of you like cured meats, like salamis. Ooh, if any of you like pickles, yogurt. All right. If any of you like beer or wine. Silence. That was the right answer. Good job, guys. Right. If any of you like bread, right? Those things all rely on fermentation. Fermentation, we'll talk about this more, produces alcohols. It also produces acids. And alcohols and acids are actually great preservatives, right? They do add flavor to some things. They also, right, keep things from spoiling. And so fermentation is a great tool, all right, that we use microbes for in the generation of some of the foods that we like, all right? Bioremediation. Does anyone know what bioremediation is a fancy word for? No? What about if you look at this picture? What do you think this picture is trying to show? I got this one, right? So bioremediation is what I mentioned earlier for an article possibility. Bioremediation is the use of microbes to remove harmful toxins and pollutants from the environment. So if any of you remember like the BP, oil spill off the coast of the Gulf, right? One of the things they were doing is actually going to those oil slicks and spraying bacteria on top of those oil slicks to kind of help break down the oil. And we'll watch one of the videos you guys have to watch is on fungi, and he actually talks about using different fungi, right, or different fungus as a bioremediation tool, right? And then digestion. So within your gut, right, you have a bunch of bacteria that actually helps you break down food products, right? They can break down things that you can't break down. And if we talk about different organisms like cows or even termites, right, they are actually using different microbes to help them break down really fibrous material, right? So that's why cows can eat grass. And that's why when your dog eats grass, it vomits it up because it can't digest it, right? You can't break down some of the things that they can. Okay. Cool. So, I do want you to know this list. I think this list is useful, and to just have a brief idea of what these things are. Be able to define them, right? If I ask what do microbes do for you and you just write these things out, well, I don't know if you know what they actually are doing. All right. So what are some of the things that we're doing with microbes? Basically, how are we manipulating them, right? And you can go back to that previous slide, and yes, some of those things are also manipulation. Some of the ones that maybe you guys think of for biotech, we can actually use microbes to help us make plastics. So some of the byproducts that they give off during events like fermentation can be used in the manufacture of plastics. Insecticides, you'll actually, one of the videos that you have to watch about fungi will actually talk about them being used to help control insect populations, specifically within people's homes. They can be used to generate fuel, all right? So again, if you are pumping gas, and most of you notice now that most of the little stickers on the gas tank will say contains up to 10% ethanol, right? So truth be told, in the United States, most of our ethanol is generated from corn, right or wrong, depending on how much you know about that. But we can also use bacteria and fungi to generate ethanol, and so they could actually be used to generate fuel to power motor vehicles. GMOs, what are GMOs?
Genetically modified organisms.
Yes, genetically modified organisms, right? So a lot of our crops are fall into this category now, and more of them are continuing to do so. And so the idea of modifying, right, or adding bacterial or fungal genes into the genome of certain plants, and so basically you can make them resistant to certain insects. You can protect them from different toxins. It allows us to grow more food. We're not going to really talk about them too much. I understand that some of you may not look highly upon GMOs. That's totally fine. There is, a lot of research that people are investigating. Do they cause, more cancer and a bunch of different things like that, right? So I'm not here to have that debate with you guys. I'm just saying this is kind of the reality of the situation and of the world that we live in, all right? So some medical advances, if you know someone that's a type 1 diabetic or type 2 diabetic, right? One of the things they have to take is insulin. Insulin used to be generated primarily from cows, all right? And one of the things that we are not is cows. And so that was kind of hard on the immune system, right? Your immune system could start to attack that insulin that was generated somewhere else. And so one of the things that we do now is we've taken the gene from humans for human insulin, and we've actually can pop it into E. coli, and we can get the E. coli to produce human insulin that we can then isolate or separate from the rest of the bacterial products. And then that's what you inject yourself with. There are also model organisms for metabolic and genetic studies. They do have differences, that is true, but they have a lot of similarities. And you can grow 1 million bacteria much more quickly than you can find 1 million volunteers for an experiment. And so things that are discovered in them, yes, eventually have to be transferred over to animal and human models, but they're a great kind of starting point to understand and study metabolic pathways and basic genetics. Okay. Yeah, we'll go a little bit longer and then we'll take a break before we kind of start doing some of the lab stuff. All right. So just a quick kind of, we'll say summary of living microbes, right? In this case, microorganisms. So back in the day when I first started school, we had 2 major domains. We had bacteria and eukarya, all right? We now know that there are 3 domains, all right? Bacteria, archaea, and eukarya. As our ability to analyze genomes, right, genetics, look at DNA of these different organisms has improved, things that we once thought were bacteria actually, you can see here, appear to be genetically diverse. And so when things branch, right, that means there's a difference. And you can see here what this implies. And again, I'm not here to argue evolution or creation with you, right? But a branch point indicates a common ancestor. So if you look at this phylogenetic tree, this point right here would be there was some organism that these all came from, right? And then there was a change and it led to these different branches, right? And then, right, these different organisms, okay? Speaking of evolution versus creation, I would love to have that conversation with you guys privately or via a Zoom session. One of the things I will mention, and I'll point this out, is that regardless of where you fall on that spectrum, one of the things that all Christians and all scientists agree on is the idea of microevolution, right? So an example of that would be bacterial resistance. When we first start treating bacteria with a drug, it kills most of them. Over time, they develop a resistance, right? Now, it's a relatively short amount of time compared to the grand scheme of history, right? So we do see micro evolution, all right? So I just want you to be aware of this, right? This is where most of the scientific field is approaching this from. And we can look at genetic differences between these organisms. Just a couple brief kind of differences is that you see archaea and bacteria are very similar in size. This is one of the reasons we thought they were the same thing. You can see a couple of these points are the same between them. And so it makes sense. Before we had modern genetic analysis, we'd look at these and we'd say they have a lot of similarities. So they're probably in the same group. You notice that eukarya are significantly bigger. It's a lot easier to see eukaryotic cells under a microscope than it is bacterial cells, all right? Bacteria and archaea do not have a nuclear membrane, right? They did not have a nucleus, right? Eukarya do have a nuclear membrane. Again, we'll talk about more of these differences. Excuse me. Bacteria have something called peptidoglycan in their cell wall. Archaea, bacteria have something that's peptidoglycan-like, but it's not peptidoglycan. We'll talk about what peptidoglycan is in just a second. You do not have peptidoglycan. Does anyone know what the cell walls of eukaryotic cells are typically made of? Anyone? Sarah, do you know? Sarah, are you there? Oh, you shook your head no? Can you hear me? Sarah, can you hear me? Looks like her screen's frozen, or she's sitting really, really still. Superpower. All right, Amanda, help me out. you say that again?
Sorry, I don't know.
You don't know. Perfect. Do you like to eat fruits or vegetables?
I love fruits and vegetables.
Okay. So what's one of the benefits of eating fruits and vegetables that people talk about, and I would say it kind of cleans you out like a scrubbing brush?
Fiber?
Fiber, all right. And another fancy word for fiber is cellulose. So we see cellulose present in the cell wall of some eukaryotic organisms, and we also see chitin, all right? Those of you that like seafood, all right, chitin is also an important component of the outer shell of many crustaceans and delicious tasting organisms, right? So that's what we see in our, well, not our cell walls, in the cell walls of our domain, right? You guys don't have cell walls. If you do, let me know, and we will study your cells and then get rich together, right? Because that would be kind of surprising. No membrane-bound organelles in archaea and bacteria. We'll talk about some of those different organelles later this week. Membrane-bound organelles. Let's see. Glory. I know I taught you some of these. What are some membrane-bound organelles? Oh, no. Wait, no, I didn't teach you these. Professor Hobbs did. Yeah, what's an organelle in your cell? Give me an example. What
I'm
makes
not sure.
What makes energy in your cell?
Like ATP.
Well, ATP is the
Yeah.
energy. What makes ATP?
Um,
What packages things? What transports things? Remember those? Those are the jobs of organelles.
Mitochondria.
Mitochondria. There we go. There's an example of an organelle, right? You're like pulling teeth. I love it though, but you got it, right? Good. Mitochondria is an example of an organelle. We'll actually see that bacteria and archaea don't have mitochondria. In fact, an individual cell, an individual bacterial cell, fulfills the function of an entire mitochondria within your cell. So we'll talk about that more. The last thing that I just want to point out is this idea of archaea and bacteria are ubiquitous. All right. And in general, we say that eukaryotic cells are not ubiquitous. There are some examples, right? I would say that fungi are actually a pretty good example of a eukaryotic cell that can be ubiquitous. Does anyone know what's the definition? What does ubiquitous mean?
Is it that they're found everywhere?
Yes, everywhere, right? Ubiquitous is everywhere. So we see bacteria, we see archaea, and you can even say fungi, right, can grow in almost every environment. So if you hear about scientists going down into the ocean and finding bacteria or some organism near a volcanic vent, if you hear about people talking about what could survive in space, right? If you heard about organisms that live in hot sulfur springs, if you heard about organisms that live in the ice of Antarctica, right? These are really extreme environmental conditions. You guys, right, pretty much need to have your body temperature around 98.6 degrees, except I did see an article now that the average body temperature has changed slightly. I haven't looked into that. Maybe one of you could have that be, no, that wouldn't be your article. If that was A&P, it could be your article. But you like a very specific environment, all right? You need certain conditions, right? Our kind of dream situation is San Diego, right? Sunny every day between 70 and 80, boom, good to go, right? We can't survive well in extreme temperatures, all right? These bacteria, archaea, and even some fungi, right, which would fall into the eukaryotic category, can. Okay? Again, just kind of a preview, right? We don't probably give eukaryotic microbes enough credit, but we will briefly talk about algae, some fungi, and some protozoa a little bit later. I think that'll be next week. Remember I mentioned the non-living things, that would be viruses, viroids, and prions. We're not going to really talk about viroids much. The only thing I want you to know is they basically are small segments of RNA, typically infect plants. We do think that there's a possibility that they're present in humans, but we haven't We really haven't isolated any yet. It's just not a major area of research. All right. For viruses, we'll talk about this more. They're really small, right? They're even smaller than bacteria. You can't see these under a light microscope. I will actually talk about an exception in the virus unit. They're made of RNA or DNA, right? When we say obligate, does anyone know what the word obligate means? Because it's actually important for bacteria too. Rachel Stock, what does obligate mean? Can you hear me? Is she frozen too?
Doesn't mean like it. What? Hello?
If
Hello?
you have an obligation, what does that mean?
Hello? Can you hear me?
I can hear you. Can you hear me?
Hello? Yes. I think my headphones are screwed up. Obligation, it means like it needs it.
Right, it needs it. So an obligate, right? So we'll talk about bacteria later with obligate aerobe, obligate anaerobe, right? It requires something to survive. And so viruses, we call them obligate intracellular pathogens. That's a fancy way of saying, all right, they can't do it on their own, right? They need a host, all right? And again, they can infect plants, animals, other microorganisms. We'll talk about these in more detail. Here's a couple that I have listed down here. I haven't updated this, right? So you could add, if you wanted to, COVID, 19. Okay. Prions, I've already mentioned with the idea of mad cow disease. There's also some other cool ones, Creutzfeldt-Jakob disease, Kuru. I have to look up the island again. I'll try to check it for you for the virus unit, which I put prions in. But basically, certain tribes on an island would eat the brains of the dead as part of the burial ceremonial celebration thing. And if you have one infected brain and you eat it and then you die, and then they say, well, you're dead, so you have to eat your brain, and it's kind of a mess, right? But really interesting stuff, right? So these are actually proteins that cause pathogenesis. So that's going to be where we stop now. We're going to take a short break before we start lab. The chemistry of life section. We're not going to go over this. I want you to look over it. This is what your review was on the Mastering My Labs platform. So if you did okay on that, and if you've looked over this and you're comfortable, then you're good to go for the chemistry of life section. What I'm going to do now, right, is sign off. So it's 9:50. Let's get back together at 10:15, and we'll go over kind of some lab basics and what you're doing there. And you guys have a very simple, I think, low pressure assignment today to do in lab via the Mastering My Lab tool. All right. So I'm going to go ahead and just sign off. I'm not going to open a separate lab Moodle page. I'll just put all of our lab information. I think my plan is to just put all of our lab information on our lecture page, since we have the same, we're all together. And that way you won't have to worry about going to two things. So 10:15, just click back on to the Zoom link for this class, right? And we'll use that for lab. All right. If you have any questions, comments, or concerns, right, please just go ahead and shoot me an email, or you can even hold on to them and you can ask me at the beginning of the lab period in about, 25 minutes. All right. Cool. Get out of here for a few minutes. We'll sign back on, and then we'll go from there. It was fun. First one down. 10:15. 10:15, Rachel.
10:15.
10:15. 10:15, Professor Perry. Well, you don't have to come to lab. I don't think that's going to be that exciting. But
Hi, Miss Perry.
Hi, Rachel. All right. I would like to be able to get on there at some point and just see what you're doing, because I don't think I've seen all, I'm sure I haven't seen all of the Pearson stuff you're going to use for the online.
yeah, and some of it's just going to be, it's definitely you know, they have a tutorial on basic safety practices. So I'm like, okay, that's good. They have a basic tutorial on bacterial culture. I think that's the first two that I have them, you know, doing today. I mean, it definitely is not ideal, but I think we'll be able to at least get them the information, you know, that they need. I wish lab, you know, was different, but
Well, I know, but I would like